Welcome to this short lecture on spermatogenesis. This lecture will essentially cover the process of how we go from a stem cell into a mature sperm. And also we'll understand the terms spermatogonia, spermatocyte, spermatid, spermatozoa, spermatogenesis, spermogenesis. So for this lecture, we're going to focus on three major images. We've got this one over here, which is a flow diagram, which just illustrates how the cells move through the different stages of meiosis. We've got, starting from the stem cell, moving all the way through down to the spermatotids. Over here, we've got an image here of the testes, more focusing on the seminiferous tubules, which we'll go through in a second. And then finally over here, this is focusing on the embryology. So how does the cells all begin with? So let's begin with embryology. Now, in this particular case, the starting of the cells to produce sperm come from a type of cell called the primordial germ cell. So going back to 2 weeks fertilization, there is two blankets of cells, two layers of cell known as the bilaminar disc. On the top layer, we have these cells called epiblasts. So there's a group of cells coming from the epiblast that migrate into an area known as the yolk sac. Now this would be the head end of the embryo, this is the tail end of the embryo, this is the amniotic sac, and then this is the yolk sac, which is going to help develop some of the intestines. So a group of cells in this region here, known as the primordial germ cells. So they migrate and start to cluster in this area. Now these cells will start at about the 4th or 5th week, will start to move back into the embryo. Now they'll go into an area around the level of T10, so thoracic 10 region, and they will go into a group of cells, like a mesenchymal group of cells known as the genital ridge. This bundle of cells, the genital ridge, essentially is just on the side, kind of medial to where the kidneys are developing, which is the pro and mesonephros. So this genital ridge is where these cluster of cells called the primordial germ cells will migrate into. They kind of migrate through like this cytoplasm membrane action where they kind of crawl their way into that particular area. And as they integrate, that genital ridge will start to differentiate into something more similar to the gonads. So when we go now, this is about the 4th, 5th week as it's doing this migration. As it starts to integrate into the mesenchyme, we'll start to have further differentiation into the stem cell, which will make the gametes. So for the female, it's going to be the oogonia. For the male, it's going to be the spermatogonia. And then we have the support cells. So as it goes into this bundle of the ridge, the genital ridge, these cells will start to differentiate slightly different depending on the hormonal situation and other signaling. So if you can imagine this part, which is going to be the testes, this is the genital ridge, and the germ cells or the primordial germ cells will go into it. This bundle of ridge is going to develop into two parts, which is the medulla cords, or the medullary sex cords, or the cortical sex cords. Now, if biologically it's going to go down the female line, it's going to differentiate more into the cortical cell lines. So this is going to develop the oogonia and the follicular cells. But for the males, this is going to go down the medullary sex cord lines, which is going to develop into these lobules. Okay, so these are lobules in the testes. So this gonad is now going to differentiate into a male phenotype. So these sex cords, medullary sex cords, are going to develop these things called lobules. So as we move towards birth, these lobules are going to number at about 250. Now in each one of these sex cords, we're going to have two major types of cells. Now, if I was to have a look through this kind of convoluted tubule called the seminiferous tubule in the whole lobule itself, so as I was saying, there's 250 lobules within a testis, and this will give you a significant volume of the testis. Within each lobule, you have approximately 1 to 4 seminiferous tubule, which you can see these squiggly lines here. A big, long tube, convoluted tube that will go up towards this end. Now, with the testes being developed, you have another duct system that comes into it. So this comes from a different system, not the genital ridge. I will explain this process in a separate video when we go through the development of the genitourinary system. But the ductal system from a separate system comes into the developing ridge, and that grows together. So this is going to be the duct or the vas deferens with the epididymis, and then the communication will come down with the developing genital ridge, which is the lobules going into the reti, testes, into the epididymis. Now, in each lobule, you'll have 1 to 4 seminiferous tubules, which is going to be this long convoluted tubule. Now, if I cut through this and look here, so now we've been born, so the baby's been born, and we're going up to a point of pre-puberty. So if I was to look and focus in on here, it would have this outer basal lamina, okay? And inside this tubule, you will have this blue cell, which is the stem cell, and these green cells called the Sertoli cells. These are probably homologs to the follicular cells in the female. So these are supporting cells, but as you can see, this whole seminiferous tubule is completely filled with these cells. There's no canals, so they have to be further hollowed out a bit later on when we go into puberty. But just for completion, remember there's going to be a whole lot of these all around each other. So all of this area is just filled with tubules because they are convoluted. And in between the tubules is the interstitium, which the cells that mostly accumulate in the interstitium are going to be things like Leydig cells and myoid cells, which is important for contraction. Okay, so when we move towards puberty, the majority, 80% of the testes is essentially filled with seminiferous tubules in these lobules. So a significant part of the testes volume is going to be in these tubules. Okay, and if you were to quantify and add up all the length of these tubules, you'll essentially probably have 250 to 300 meters worth of seminiferous tubules. Now, before we go into the actual development of the cell, I'll bring you across to this particular flow diagram to show you how it actually occurs from a very simplistic point of view. So this whole process from a stem cell all the way through to a spermatid, okay, which is kind of an immature sperm, to the point where we have sperm that's now mature, this whole process is known as spermatogenesis. However, the last part of it, where we go from a spermatid to a mature sperm, this is known as spermogenesis. So let's start at the beginning. So here we have a stem cell, which essentially started off from the primordial germ cell. So this stem cell is sitting at the basal lamina, the most lowest part within the Sertoli cell, which I'll get to in a second. This is the stem cell that makes all the sperm. Okay, so it essentially will replicate into what we call a dark type A spermatogonia cell. Okay, so the reason why it's called dark is because the way the chromatin, so the way the nuclear material is arranged, it looks dark in histology. Now this cell will essentially do two things. It will just go through mitosis and then just go back on itself to continually create a pool of cells that will always be there for the production of sperm through the whole age, the whole life of the male. So we do have a population of the type A cells that just keep going in this circle. But there is a group of type A's that will differentiate into what we call a pale type A into a type B cell. So these are now essentially going into a type B spermatogonia, which has 46 number of the DNA chromosomes with an XY. So it's got double the material essentially as a normal somatic cell. Now this particular cell will then migrate into what we call a primary spermatocyte. So we're now ready to essentially go through the first stage of meiosis 1. So this is going to be meiosis 1 from the primary spermatocyte into the secondary spermatocyte. So this is the largest cell out of the whole lot. So through this whole flow process, the primary spermatocyte is the largest out of the lot. Now, the primary spermatocyte will break into 2 secondaries through meiosis 1, and so you're basically halving the amount of genetic material. So with the secondary spermatocyte, essentially what we do now is halve it again, and so we split it through meiosis 2, and now we've gone from a diploid into a haploid, and then we split again into another most simple form, and these are what we call spermatotids. Okay, so essentially from one cell, we go into 4 spermatotids. So that's essentially the process, the flow process of how we go from a stem cell into spermatids, and then we're going to go into maturity. But what we're going to do here is now jump into the actual testes to see the process actually occurring. So as I said, if you were to look at this in the pre-pubertal stage, these seminal nephros tubules will be all filled with these types of cells. The most abundant cell will be the Sertoli cell, and you're going to have the other type of cell within the tubule being the germ cell, which is going to be that one. But as we hit puberty and the hormones start to change, things like follicular stimulating hormone and luteinizing hormone, which is going to come up from the pituitary gland, come down from the pituitary gland, it's going to have an influence on 2 major cells, one being the green cell, which is the Sertoli cell, and one being outside here, which is in between here. So it's going to be sitting out here, being the Leydig cell. So here we're going to have a change in testosterone, and the way that the Sertoli cell, which you can see is arranged here, it's going to now arrange itself like so. So if you could imagine this basal lamina is going out around like this. So I couldn't draw it all, but you could probably imagine that this would continue around in a big circle. And in here in the middle is the lumina, which is the canal where the sperm is going to move through. Okay? So the Sertoli cell, kind of, as it goes into its adult form, will start to look more like a kind of a columnar or more like a cuboidal shaped cell. But a lot of this is the nucleus, and the way that the cell arranges, this is kind of like a plasma recess. So this is a whole lot of tissue, or a whole lot of cytoplasmic tissue that the developing sperm is going to be nourished within. So one of the primary functions of the Sertoli cells is to actually nourish the cell as it moves through into its mature form. It's also going to play around with some hormones, particularly the hormone called ABP, which is androgen binding protein, but it's also going to produce another hormone called inhibin, okay, which is probably going to feed back up into the pituitary gland and change the speed or the way that this spermatogenesis occurs. And also it's going to produce a fluid to help move the sperm along the tubule up towards the epididymis. So now we're going to focus on the process of how we go from a stem cell and go and produce a mature sperm. So down here we've got the stem cell. So this is the stem cell spermatogonia cell, which is this one. It's going to replicate like so, and it's going to produce a dark type A cell like that, like this one. This will essentially feed back, and this process will just continue to maintain that pool of cells. But some of these type A cells will differentiate into a type B cell, which is going to be here. This will further, through mitosis, copy itself, okay, and then go into a primary spermatocyte. Now it's important to note here that the Sertoli cell has kind of 2 parts to it. It has a basal part and it has an adluminal part. Now there is kind of like a tight junction between the two, which is kind of like a blood testes barrier, which is important for immune function to stop the immune system essentially attacking or possibly interfering with this process. And the important point here is as we move from the primary into the secondary, you have to kind of move through that tight junction, or that area where it's kind of inhibiting the communication between the two. Also, the setoles neighbouring each other have other tight junctions which seal them off, which also allows that blood testes barrier. From the primary spermatocyte, which we're here, as we said, we go through meiosis 1, which produces 2 secondary spermatocytes. So we've gone from spermatogonia now to spermatocytes. From here, we then halve again and we go into the spermatids. As we move towards the lumina, the luminal region, so the canal of the seminiferous tubule. So this is going to be in the middle part to be transported up into the epididymis. So we're really in this point here, which is now spermogenesis. So the process that I'll go through quickly is now how do we go from an immature spermatid, which is this particular cell here, which would be these ones, into a mature sperm. So there's a few steps that occur here. So as we've copied into the spermatid, which is here, we have to do a few phenotypical changes to the cell to make it ready to be an active sperm. One of the first things it will do is the Golgi body will produce these kind of lysosomal bodies, which are enzymes. And these enzymes is going to be the acrosome. This will start to arrange itself kind of in the front end of the nucleus. So the front end of the nucleus, which is important to be the head end of the sperm. Now what further happens is the cytoplasm starts to pinch off, which gives you the appearance of the acrosome will sit at the front end of the sperm with the nucleus behind it. At the same time, we start to have these protein-like contractile units that will start to come off the back end of the nucleus and go towards the tail end. These are going to be, now the membrane itself starts to pinch off and go dorsally, and this is like cilia, but it will eventually go into a flagella. These protein-like contractile elements, which there are 9 in number, start to go paralleling that part of the cell. And we also have mitochondria that start to increase in number and accumulate just behind the nucleus, which is going to be the mid-piece of the sperm. And the mitochondria is going to be important for producing the energy to propel the flagella and move the sperm along. Finally, what we see happen is the excessive part of the cytoplasm, which is this part here, will get phagocytosed and taken up back by the Sertoli cell, and then the sperm itself, or the spermatozoa, will detach from the Sertoli cell and now sits in the lumen. From here, the fluid that the Sertoli cells make, which is a protein-rich fluid, will start to excrete and push the sperm along the seminiferous tubule. As it moves up towards the rete testes, the cell type starts to change into more of a cilia type, and this can propel it along into the epididymis. At the same time, as we move kind of in this region, all that fluid is reabsorbed back into the testes, and the actual ducts itself is very high and rich now with spermatozoa. So this will now not so much mature the sperm, but actually now make it motile. So as we move into the epididymis, what the changes in from this part into the more proximal part is going to be essentially the sperm become more motile, and then when ejaculation occurs, it goes up the ductus deferens up into the abdomen for ejaculation. So hopefully now what you've seen is how we go from a stem cell in the embryo, okay, we produce the testes with the seminiferous tubules in the lobules, which give you the vast amount of volume in the actual testes. How we go from a more of a primitive structure of the seminiferous tubule to more the adult structure with particularly a strong focus on the Sertoli cells that's supporting. The Sertolis are also called subsentacular cells, which, as I said, is probably the homologue to the follicular cells, which helps to support the germ cell. And we've seen how this germ cell changes from a spermatogonia into a spermatocyte, into a spermatid, into a spermatozoa. And that process, all that process is spermatogenesis, but the latter process is called spermogenesis.